Thank you so much for being here.
Sure, of course.
So first, I was hoping you could just provide kind of an overview. What is H5N1? How likely is it for humans to contract this virus?
Right. So H5N1 is really a novel strain of influenza virus. The flu virus uses two proteins in the way that it gets into cells and how it's comprised. And so it can switch kind of the ability to, one component is called H, one component is called N. And so each of those numbers represent a different format. So seasonal flu typically has a very conserved H protein and N protein, but they do change, which is why our seasonal flu vaccine every year is slightly different because there are modifications. But this is considered a much bigger change. And so it's likely that it could cause a disease that is significantly worse than seasonal flu simply because we as humans don't have immunity to that particular strain.
Right. We haven't been exposed like we are to seasonal flu.
Exactly. We don't have vaccines that we had over time and build natural immunity and also immunity that comes from vaccination.
Right. Exactly. So at this point, how likely is it for H5N1 to spread to humans? Where is it circulating
right now? It's primarily in animals. It's in many different animal species, and there is animal to human transmission, which means that for people who are around infected animals, for example, our cattle supply in the US, those workers are seeing a lot of influenza virus. And it's also not clear how one would necessarily be able to avoid that if that's the sort of work that they do, because it's impossible to completely protect yourself from the transmission event. What has not happened yet, and what I think we as scientists are, hoping will take longer, is human-to-human transmission. And so that is really the point by which there will be another likely change in the proteins of the influenza virus itself. And that is, I think, what causes us great concern, because that then means that would be a big enough change which would stop seasonal flu vaccines and even antibodies or vaccines people are making to the current H5N1 construct will likely be much less effective.
Right, because it'll be that much more different.
It'll be that much more different. And then once that happens, then the probability that we can use vaccination of existing vaccines or antibodies of current circulating strains will simply not be very effective. And we saw that with COVID-19. We saw that the first monoclonal antibodies that were generated both here at Scripps and throughout the world, pharmaceutical industry took a massive undertaking to build monoclonal antibodies, that as COVID-19 changed, those antibodies essentially became ineffective and got pulled.
Yeah, just completely evolved past them.
Exactly.
So what are some of the current efforts that people are working on to help mitigate the spread of H5N1 before it gets to that kind of pandemic potential?
Yeah, so I think there's a lot of questions around what surveillance looks like, right? So as we know from work, not only at Scripps, but also at other institutions and public health authorities, that looking for the virus, be it in water supplies or in other, animal, transmission events and whatnot, that has been the primary tool that we've used. The challenge with influenza virus is that it is exceptionally widespread now.
Right, it's so pervasive.
It is likely in many animal species that we simply don't track at all.
Yeah.
And when that starts happening, then the likelihood of another mutation leading to transmission between humans becomes more likely. We saw that in the context of COVID-19, but certainly in flu, it's already exceptionally well precedented.
Exactly.
Previous pandemic flu viruses underwent a similar sort of pattern where it spread very widely amongst animals, and then it took sometimes a time, but I think it's important time that we have. So right now, surveillance is a big part of it. The other part that's a big part of it is trying to build vaccines that can be used against multiple H proteins and multiple N proteins. And so that is early stage, and people are looking at various different approaches. The NIH is doing a lot of work, less so within the private sector, but the majority of it's within academic institutes and the government. But finding pandemic flu vaccines has been a big challenge. And we don't have anything right now that is available. So, what we would be left with is the ability to generate a vaccine once there is a well-characterized strain, which we saw similarly to SARS-CoV-2 and COVID-19. and that will take some time. And I
Yeah,
think that's
absolutely.
that first few months. Let's just say optimistically, though for COVID-19, it did take 6 months until we identified the virus and we had the first vaccines available for larger populations.
Right. No, that makes sense. So, what were some of the other key learnings from COVID-19 that have prepared us for this? I mean, obviously, you touched on surveillance, which I'm sure a lot of that infrastructure in place now is hopefully able to be used. I understand that it's a much more pervasive virus, so it's harder to track. But yeah, what are some of those key learnings we have from COVID that can be applied?
Yeah, I mean,
Hopefully now.
certainly all of the mitigation strategies to protect vulnerable populations, right? Everything from being able to limit contact or to at least have proper protective equipment, certainly in the hospitalized setting, is critical. But I think one thing that we didn't learn from COVID-19 that unfortunately will be much more pervasive with the pandemic flu virus, based on what we know from previous pandemic flu strains, is protecting young people and protecting children. We were able to get away with COVID-19 in many ways, opening schools earlier and being able to have adult to children and children to
Right.
children contact that did not allow COVID-19 to spread. So I think that is going to be likely the biggest impact. I think
Right.
vulnerable populations, those who are immune suppressed, those who are elderly, have comorbidities, those folks are certainly going to be challenged with flu
Right.
like they were with COVID-19, and we see that with seasonal flu. But what we have not seen is a pandemic flu virus and the effect it can affect children. So I think
Right.
that
That's
that is
true.
one area where a lot of the mitigation strategies that we had that got by for
Yeah,
COVID-19
just
will
would not
simply not be
apply.
enough,
Yeah.
for a pandemic flu
Yeah,
virus.
that makes sense. Yeah, that I guess was one of the bright sides of like the COVID pandemic was that it didn't affect children the way that past seasonal flu pandemics have.
Exactly, yeah. So pandemic flu viruses, Spanish flu virus, many young children were affected and high mortality rate. So,
Right.
so within that context, I think, the development of the other thing that, I wanted to mention too, we have learned a lot about how to develop drugs for influenza. And I think that those sort of learnings are now starting to make their way. NIH has become much more accepted. WHO, other authorities around the world are starting to recognize that we need more drugs to deal with the flu
Right.
virus.
More of like an arsenal behind.
Exactly,
Yeah.
exactly. And those things are beyond, what seasonal flu kind of does and can lead us to pandemic flu preparedness. But it's still in its infancy. I think, if we look at the history of COVID-19 drug development, the drugs were really not there. Remdesivir was for other purposes, molnupiravir
Right.
was for other purposes. So we had to kind of do bottom-up drug development. I think we're in a much better situation now for another coronavirus.
Right.
I think we have many more tools, but unfortunately, our tools for influenza, which is a very different virus, it does not encode similar proteins
Right.
as coronaviruses. The only thing that is in common is that it's spread via respiration,
Right.
and so those parts are, we're underprepared for that.
Okay. So, what are some of the things kind of in that arsenal that are helpful? So, you mentioned drugs. So, I assume antivirals. Why are antivirals also necessary if people are developing vaccines,
Right.
in tandem?
Right. So the reason why antivirals are important is because they affect the proteins themselves that the virus encodes, right? So a virus encodes only a handful of genes versus many genes that mammals and even eukaryotes generally do. So what we've learned is that when we use those, when we have those limited targets, we really don't have as many different ways to treat someone who has an influenza virus,
Right.
but there are proteins that the virus needs to be able to replicate. So when we're talking about how do we block the virus from getting in, we have those antibodies and we have the vaccines. But the challenge is that over time, the virus, like it did with SARS-CoV-2 and COVID-19, will evade those mechanisms
Right.
and come
Mutates
and
very
come up and,
quickly.
and come up with new mechanisms to be able to get inside of a cell. The advantage of antivirals is, and especially direct acting antivirals, so ones that affect the actual replication of the virus.
Right.
Certainly there are drug targets that one can use to stop the virus from getting in, and some of those are actually conserved mechanisms, so you would be able to hopefully be able to avoid resistance
Right.
and
Mutating
avoid
this quickly.
mutations.
Right. It would
But
last for longer.
exactly, but once it gets inside the cell, then it has to use those proteins. And if we come up with effective combinations of drugs that stops the virus from mutating against one of those targets, we also have been exceptionally fortunate with COVID-19 in that respect.
Right.
Paxlovid, remdesivir, molnupiravir, they don't appear to cause mutations. The virus does not appear to want to mutate away from those.
Right.
Unfortunately, for flu, we already know for seasonal flu, the drugs that have been developed for seasonal flu, that there's quick evolution and rapid mutation of the flu virus towards those drugs. So we need to come up with interesting strategies of how to combine drugs together so we can limit that. And we know from HIV and from hepatitis C that the combination of drugs has a profound effect on improving efficacy, but also profoundly improves the ability for the virus not to mutate away from those drugs. And so I think that's kind of the call to action.
Yeah, more of that cocktail approach, so it can't really circumvent anything.
Yeah, and we didn't have to do that for COVID-19. And we joked about it a lot, that there was no scientific need for Pfizer and Merck to work together to combine Paxlovid and molnupiravir, for Gilead to work with Pfizer and Merck. I think for flu, we will need to have either people working together to do it, and in a world where they don't, we'll need to have the nonprofit and academic world to be able to move forward in drug combinations. I think that's a critical aspect.
Right, and this larger collaboration component, right? All of these sweet spots, academia, smaller biotech, larger pharma, all have their role to play, right, in making sure that we can actually develop these quickly, effectively, and share as many resources as we can.
Exactly. Yeah. And I think for flu especially, we'll need to be mobilized. And so now we have some hints of what we can start doing today. So I think that's really critical to get that process started now.
Right. So what is Scripps Research doing to combat H5N1?
Yeah, so obviously I mentioned the universal flu vaccine approach. So there's multiple groups here at Scripps who are working on universal flu vaccines. It's really exciting work because I think that that's really going to be a critical element in this. And then what technology do we use to kind of manufacture those? So that's one component of it. Understanding the structural biology of those proteins that the virus uses to get inside the cell. Tremendous amount of expertise here to be able to understand what those, at an atomic level, what those proteins are, how we know seasonal flu. For example, Ian Wilson's group does that, and understanding how the seasonal flu changes and how we can, on a molecular basis, design better vaccines, better antibodies. And then at Calibr, working together with several folks, faculty members at Scripps, working on combining and coming up with new antivirals. So those would be ones that we can produce now. We can stockpile today. We know that they will be effective if used appropriately. There's been a larger interest in anti-infectives of what's called anti-infected stewardship. And really, a lot of this comes from clinical practice, right? How do we effectively use drugs in a hospital setting and even in an outpatient setting to be able to limit drug resistance? And I think that has actually been quite successful just in the last few decades of how physicians actually prescribe and maintain the safety of drugs and the efficacy of drugs as long as possible. But I think we need to start coming up with drug combinations for influenza. We're doing that at Calibr. And what's really exciting about that work is that that's something that can be cheaply made today. It can be stockpiled. It can be made available as soon as there is any examples of human-to-human transmission. And even in the context of COVID-19, even though we didn't have those tools, people now say that if there were those sort of tools available, that one could give to high-risk individuals, for example, people who could succumb to the disease, but also to healthcare workers and other folks who were involved, that would have dramatically impacted the spread and allowed the vaccines and the antibodies and all the other things that needed to be developed only once the pathogen was actually sequenced and understood, could we actually start, having a much bigger impact and limit the amount of casualties.
Yeah, absolutely. Yeah, you mentioned healthcare workers, people that are on the front lines and just so exposed to these viruses. So yeah, it would have just been so amazing, especially in those early days, to have something stockpiled, as you mentioned.
Yeah,
that would be easily accessible.
And I think that a lot of it also needs to come from, the ability for us, and something we've been very mindful of is, how can we make these drugs cheaply? How can we have a lot less drug that we need to make? How can we maximize the number of doses that can be given to people, not only to, if they have infection, to not succumb to the illness, which, just to remind everyone, it's 60% is the historical average of pandemic influenza virus. So it is not the single digits that we know happened with SARS-CoV-2 and COVID-19. So in that world, we need effective treatments that can be taken early. And we also need ways to be able to protect people who likely are going to see a lot of virus. And so both of those things really come from the same set of tools. So the one nice thing about direct-acting antivirals is that they can be used not only for the treatment of the disease, but also to protect people from getting the disease. And we know that exceptionally well from our work in HIV and in other unrelated viruses, but similar transmission.
Right, exactly, where you're having to, like we mentioned, more of this arsenal cocktail combination approach.
Exactly.
So I also want to talk about Reframe as well, because that was a huge assistance in the COVID-19 pandemic and identifying potential drugs that could be helpful. So can you explain what Reframe is and how it's being used in this case?
Yeah, so Reframe is essentially every small molecule drug that, again, can be produced cheaply, made available, stockpiled, and make them available to the entire community to test. And so that actually is a pretty large number of molecules. It's about 14,000 molecules.
Yeah.
And what's really amazing is that if you went and asked the question, how many of these molecules can I buy and test? It's only half. It's actually slightly less than half.
Oh, wow.
And so we actually, with the Gates Foundation, undertook a massive campaign to make all of the molecules that you can't buy and make them available in quantities such that we can do many, many screens, both in vitro and in some cases in vivo, interestingly enough, to be able to provide those to the scientific community to test free of charge. All the only requirements is that they need to publish the data so that people can review it and take it available.
Open access.
Exactly. And so we did that in January of 2020 with SARS-CoV-2. We found a lot of molecules like remdesivir and other things that were effective. We found many things that people claimed were effective that at least did not work well in our hands, and didn't work well in our hands, meaning it didn't work in enough systems for us to actually say this could potentially be useful for treatment of COVID-19. And so we've made that tool available. We've also used it and tested it against flu, and we found interesting molecules that one would need to develop further to be able to get to an interesting molecule to use for H5N1.
That could actually be effective in humans.
Exactly, and so that's the work we're doing in the lab right now. Certainly, we've learned many things about it for COVID-19. We've used it against other viral infections. We've used it in other respiratory diseases like tuberculosis, things that aren't even viruses.
Right.
And the beauty of it is that it allows the community to access it. It allows a platform on reframedb.org for people to compare their data to other data that's been generated in similar systems. So you could start getting a confluence of data that suggests that one molecule may be more important to follow up and actually look at in more detail. And so that's what we've done with flu, especially over the last 2 years, to really come up with things that not only could be useful against seasonal flu, but also against pandemic flu viruses, again, accessing and hitting those targets that are conserved between the different types of influenza viruses. And so that's kind of what we're doing in the lab right now. So, it's an amazing opportunity to provide those molecules. I'll say we have a lot of work left to do on that front. There's still about 2,000 molecules in Reframe that are not commercially available that we have not made yet. And so I think that's an opportunity for even, in a more meaningful way, increasing the amount of data that we have to make sure we're not missing anything.
Yeah, I think covering your bases. If we already have all of this information, we need to make sure we're utilizing it.
Exactly. And I think that's the real challenge, is that, as a scientific community, we need to be able to do these things proactively and be able to make the data available and allow people to critique the data, for people to come up with other ways to test. And so that's been really exciting to be part of. And I think that will only have greater and greater traction, and for people to be able
especially as more and more people use it, right? Yeah, just kind of the exponential growth of that.
Exactly. We're constantly talking about, I think, in a world now where we're talking about, how to integrate in together large amounts of data, and how do we use that? How do we use that in appropriate models for us to think about how can we actually design and come up with new hypotheses and new things to test in the lab. I think, it's a real shame that, we have half of the number of compounds that have been safely put into humans that we actually have very little data on. And certainly the people who develop those drugs have a fair amount of data, but what's in the public domain is actually
It's very,
quite
very
small.
limited. Yeah,
And so to really be
absolutely.
able to build out that data set and to enable that in the Calibr
Yeah, it's
Scripps
amazing.
ecosystem is
I
fantastic.
think that's an incredible caveat. Yeah, where it's you can access this, of course, but you have to, yeah, freely publish
Exactly.
whatever it is. Yeah, it's incredible. So for everyone listening, what is your call to action to the community to help mitigate the spread of H5N1, or kind of what can people do now, regardless of kind of who they are?
Yeah.
Yeah.
I mean, there's a lot of things to do. I think, what we learned from COVID-19 is that, it touches everyone's life in a profound way. And I think in many ways, we can understand that there are some things that we can do, right? Personal protective equipment, limiting interactions, but we know that over time that has a significant impact on society.
Yeah.
It has a significant impact on our mental health. It has a significant impact in our economic health. It has a significant impact in people's ability to live, their lives.
Right.
And so from my perspective, I really think the call to action is be aware of kind of what's going on, in terms of public health. I think our public health infrastructure more and more needs the support and recognition from everyday citizens around, how is something spreading, and not necessarily in a doomsday kind of panicky sort of situation.
Right.
We were with COVID-19, we were kind of left in that situation.
Yeah, right.
And so now it's really about, okay, we do know a lot about seasonal flu. We have learned a fair bit about pandemic flu. So it's not as this an insurmountable challenge for us to be able to come up with an appropriate pandemic preparedness response, but we need to be aware of kind of what's going on. And I think part of that also involves educating those, who may not be aware of those things. And I think COVID-19, unfortunately, has polarized people in terms of how they think about mitigating it, but I don't think anyone would argue, at least thoughtfully argue, that it didn't have a huge impact on us.
Absolutely.
And there was not a single country in the world, no matter what their strategy was to deal with the virus or mitigate it, that was not affected, in a profound way. So I think that is an essential thing we need to understand. I think, for me, it really does come down to being able to invest in the best science to be able to make sure that we're better prepared for pandemic flu than we were for SARS-CoV-2. And I think that does come down to us being able to say, it's an important investment. It is an insurance policy against society functioning properly. And there's a lot of need for us to be able to get the best minds, but also get the best resources to be able to have that impact. When you're talking about a 60% mortality rate, you're talking about a disease that when it affects someone, we need to act very quickly.
Absolutely.
And so, what we've learned in the context of Tamiflu or seasonal flu is that, drugs work okay. They're not given necessarily early enough. We don't always necessarily have the right means to apply it, but those drugs do work and they do stop many people from succumbing to the disease. And so we need to be able to understand that, onset of an illness, especially in vulnerable populations, requires immediate action. And I think we need to, as a society, do a much better job of educating people, but also be able to train them, and then most importantly, allow them to act, to be able to come up with interesting antivirals, interesting vaccine designs. And for me, I think you need both, because those first 100 days, there'll be a lot of panic, right? And there will be a lot of people wondering, how is this going to affect people?
Exactly.
And we can't ahead of time predict all of those aspects. But if we have the right tools in place, understanding where the disease is, being able to treat and prevent people from the disease, and then accelerate manufacturing of vaccines and other things that will have even a greater impact to protect people from the disease, I think is essential.
Right. Amazing. Okay, I don't have any other questions.
Right.
Is there anything else that you think would be important to cover that we haven't talked about?
Yeah, so I mean, I think one of the things that comes up in my mind often is the need for collaboration, and I think that no one organization can do this effectively. Reframe is just one such example of that, but I think working together with the federal government, working with people who are very good at drug manufacturing, and being able to say, okay, we have something very interesting here. How do we scale this? How do we make it available to enough people around the world in centers where we think that, there is, a greater impact of the disease. I think that's a critical element of it. So my hope is that, and obviously we do a lot of that with the Gates Foundation. We do a lot of that, in the US, we do that with WHO. We do that with a lot of the work that we do in other parts of the world. But organizing all of that in a collaborative way and having the right platforms to actually share data and share information and act quickly is something that I think we can continue to strive for. And I think in COVID-19 we got a little siloed, right? At the end, people were developing drugs and developing vaccines, but they weren't necessarily sharing data and communicating. And I think we'll need to certainly do that in this case, because I think this will be one where there is a lot of foundational knowledge around influenza. The world of coronavirus, folks,
it was brand new, but
it was a very, very small group number of people.
Yeah.
And in flu, we luckily have many, many people who've done really great work in flu, both in the private and in the public sector. Bringing those folks together, I think, is going to be essential. And so I think that's one of the things we're excited about, is being able to do that. And certainly there's actors like the Gates Foundation, NIH, and others. But I think as an institute that's doing science in the lab, training the next generation of scientists, and then making these impactful, drugs to put into clinical trials, I think the collaborative environment is really critical.
It's absolutely key. I mean, I can imagine it must be so challenging. The organizational structure of all of these different entities is so different. Yeah. And so to be able to communicate, share things collaboratively, I can imagine would just seem insurmountable in certain cases.
And I think we can start that now.
Yeah.
I think that's something that we can do now. And we've had a lot of great discussions on kind of, how to generally deal not only with pandemic flu, but with other viral and non-viral pathogens that are communicable and are ones that will affect people very quickly if not properly dealt with. And so I think that process has started, but I think, as people are looking perhaps maybe a little too siloed in their own countries and their own states and in their own regions of the world, that I think that's a force we have to kind of push back against.
Yeah.
And I think the academic environment and I think the nonprofit sector is the best place to actually
have that kind of collaborate, like open communication and collaboration. Yeah. Exactly.
Yeah. Great.
Well, thank you so much.
Thank you. It was fantastic.
Yeah, no, that was great.